Hey guys, so today we're going to be going over the key differences between meiosis and mitosis. I know they sound very similar, so it can be slightly confusing, but we're just going to make a little T-chart comparison that tells us the differences between meiosis and mitosis. So the first thing is what they're producing. Meiosis is producing 4 haploid, remember haploid is half the chromosomes, unique gametes. And remember, gametes are sex cells, so that's either egg or sperm, egg for female, sperm for male. Mitosis, on the other hand, produces 2 diploid. That means it has all the chromosomes, identical somatic cells. Somatic is basically a body cell. Basically any cell in your body that's not an egg or a sperm. That's every other cell in your body. So that's your skin cell, that's your heart cell, that's your liver cell, that's your brain cells. Any other cell that is not a sex cell is considered a somatic cell. Okay. The main purpose of meiosis is to produce these gametes, but also to increase genetic variation. For mitosis, its main function is for growth and repair. So, for instance, growth, like this is how you get taller, this is how you get bigger throughout your lifetime, from literally the very beginning. So we talked about fertilization where egg and sperm come together. When the egg and sperm come together, they create a zygote. The zygote then turns into an embryo. Embryo is like the beginning stages of a child. The way that you're able to grow that embryo is through mitosis. Mitosis just produces essentially clones of the original cell. Mitosis does not affect genetic variation. Because it is a form of asexual reproduction. Meaning it creates clones of the original cell. Whereas meiosis is part of sexual reproduction, and that's why we get all of this genetic variation. There's also a couple differences in terms of the phases that you go through. So in mitosis, we know that our phases are prophase, metaphase, anaphase, and telophase. Those are the only 4 phases for that. In this prophase, in mitosis, there is no crossing over. Similarly, in metaphase, there's no, like, the independent assortment. Like there's no independent assortment, it wouldn't matter. In meiosis, you have basically 2 rounds of division. So you have prophase 1, metaphase 1, anaphase 1, and telophase 1. These little dashes are 1s. And then you have prophase 2, metaphase 2, anaphase 2, telophase 2. Now the main difference between these is that in the first round of division, you have homologous chromosomes. Like anytime in these sets you're seeing homologous chromosomes go through this phase, remember homologous chromosomes looks like kind of 2 X's. Whereas in the second round of meiosis, you are seeing sister chromatids. And remember, sister chromatids just look like 1 X as you're going through the phases. That's one of the main differences of how you can tell if you're in the first round of meiosis or the second round. The other key thing I mentioned, specifically prophase, that no crossing over occurs. Well, one of the main ways that genetic variation occurs is in prophase 1 through crossing over. So those are the main differences of mitosis and meiosis. I'm going to zoom out so you can actually see this. And make sure that you continue with this practice. You're going to be sorting the statements below into meiosis or mitosis, and you'll continue through the packet by following your checklist. Let me know if you have any questions and have fun.